Okay, I think it's far enough past the top of the hour to start. Welcome everyone to this week's viral pandemic working group seminar. James Glazier is out of town in Paris and is having some trouble connecting, so I'm going to run the meeting today. Try to run the meeting today. We'll see if we can get everything all sorted out. Let's see. So our usual set of slides. All of our people, Tom Helicar and James Glazier, the two leads of the group. Myself and Lorenzo Vicini are the help coordinate some of the nuts and bolts of the system. As does Joel Vannon at IU. We have various social media accounts. There's our Slack channel, Twitter, IMAG, MSM wiki page, LinkedIn, and YouTube. Hopefully this talk will be up on YouTube in a few days. All of our previous talks are on our YouTube channel. We have a Twitter line. If you get a tweet from us or see a tweet from us, please hit the retweet button. Try to expand our audience a little bit. A little reminder that the Building Immune Digital Twins meeting in Paris starts Monday, which is why James is in Paris and a lot of other people in the group. And that might be why our turnout today is a bit low, because so many people are at this meeting. Hopefully they'll get very exciting things done, and hopefully we'll be able to get a seminar from the output of this meeting on this channel sometime in the near future. Reminder that the IMAG MSM meeting is coming up end of June in Bethesda, Maryland. I believe the abstract deadline is tomorrow. So if you're interested in getting an abstract in for that meeting, for a poster for that meeting, you're running out of time. Quick reminder that IU, Indiana University, has two open professor twins in the Intelligent Systems Engineering department. Contact Professor Glazier or check the posting at indiana.peopleadmin.com. In addition, there's multiple faculty positions at the University of British Columbia. You can check it at isi.ubc.canada. Finally, I think this is our last one. Nope, almost. The GLIMPRINT site is up. If you're interested, give it a look. If you'd like to join, there's a join link. We're working on a new logo, but fair amount of the total website is at least up and functioning at the time, for the time being. Upcoming seminars next week, or actually 2 weeks from now, the 25th. There's no meeting because so many people are at the Paris workshop. Following that, on June 8th, we have Megan Haas at the University of Virginia, Multiscale Modeling of Western Effects on Muscle Regeneration. And then June 22, Harm Public Health in the Netherlands, Multiscale Modeling of Toxin-Induced Spinal Tube Defects. If you have any other suggestions for speakers, feel free to use the form listed there. We can always use more speakers. Okay, so I think we got all the background stuff done for today. So I'll just jump right ahead and let David start his presentation on modeling the ecosystem in cancer with agent-based modeling.
Well, thanks, Jim. Thanks, James. Thanks, Lorenzo. It's great to be here this afternoon. My name is David Basanta. I'm a senior member at the Moffit Cancer Center and professor. And for those of you who don't know, the department I belong to is called the Integrated Mathematical Oncology Department, which is a very unique department inside a cancer center. So we are a bunch of physicists, computer scientists, mathematical people who work inside a place where usually experimentalists and clinicians work. So it's a very interesting environment. This is an old picture when people could get together in close confines and be maskless, but it's the only one I found. So this is a picture of my group. It's always important to acknowledge the people who actually do the work before we reach the last slide, which we might or might not be arriving to today. obviously, I was thankful to the funding as well, one of which is IMAG and the MSM initiative itself, which funded one of the projects I might be able to talk about today. So being a cancer-oriented group, we obviously get funding from the NCI as well. And obviously, one of the big things about the work we do here at Moffit and in IMO and in my group is the fact that mathematical modeling is important to us, not just because we really care about the math models we produce, but also because we believe they are the glue that integrates insights in terms of the biology, data in terms of the clinical data generated, put together in ways where we can actually not only create interesting models that are interesting in itself, but also because we can actually use those models to guide our understanding of cancer, and very importantly, our understanding of how treatments are applied in the clinic. And to do so, we actually work very closely with clinicians and experimentalists. In my case, though, my closest collaborators tend to be experimentalists, and the people I'm going to be describing, whose work I'm describing today, include collaborations with you see on the left, the group of Connor Lynch, which is a bone biologist here at Moffit, and on the right, the group of Andrei Manusik, who is a cancer evolution specialist. And this is basically one of the few pictures I got on Google about prostate and prostate cancer. Most of them are not suitable for work. I know that most of you don't necessarily work on cancer. One of the beautiful things about going to MSM meetings is that you get to talk to people who do multiscale modeling on all kinds of diseases. And being in a cancer center is great from the point of view of actually understanding the disease very well, but it limits you to the problems people have in terms of health. There's no heart problems, there's no skin problems, it's mostly just cancer, right? So I'm going to be showing a number of models, and some of them are not new and some of them are really recent, and I'll see if we actually go and cover them all, but I just want to go through some of them with the idea that sometimes you actually gain to learn a lot. Not by just looking at a particular model, but by putting them together and seeing where they can actually tell you something that one single model itself wouldn't. Now, I know that we have a relatively small audience today, and I would encourage every one of you to please stop and ask questions at any point, because I don't think my goal here is to just basically describe a bunch of models. My goal here is to start a conversation about how math modeling can actually, multiscale math modeling to be more precise, can actually help uncover how the interactions between disease and the host, trying to be as generic as possible, can actually explain how the disease progresses and actually gives us new venues by which we can prevent that disease progression. So please do not hesitate at any stage to stop me and ask questions, because just having the conversation is going to be a lot more important than just going over the slides. That's not even going to be the goal. So when it comes to prostate cancer, there's a number of stages that determine, actually, I see that there's something on the chat. Okay, no, I'm sorry. I guess that I was expecting some new messages, but yes, if you actually have questions, it's probably best that either you use the chat and maybe hopefully Jim can actually interrupt me and ask the question, or you just use the microphone and ask the question directly. As I was saying, one of the things you can tell about prostate cancer is that there are many different stages of progression in prostate cancer. Now, you can look at them from the biological perspective and you look at genetic transformations, but if you're a clinician, usually what you look at is the stages in terms of the physiology of the disease and in terms of how the disease looks under a microscope to a pathologist. So the early stage is basically the disease initiating itself and going inside the prostate glands. Invasion is when they break from these glands and start invading the entire prostate. And metastasis is when the disease goes from something relatively benign to something that is life-threatening. So let me just get started with this. If you are a cancer biologist and you are a clinician, you look at this and it doesn't really ring like anything that you know of in terms of prostate, but if you are a mathematically inclined person and you feel like you can use circles to describe prostate glands, then this is how a prostate gland looks like. They are basically these structures that are basically spotted all around the prostate, and they have these two layers. made of epithelial cells. Most cancers start from epithelial cells. So 80% of the cancers are carcinomas, which basically that's what it means. They start from epithelial cells, lots of cells in your body, but most of them don't lead to cancer most of the times. Epithelial cells are a lot more likely. And you can see these two layers of epithelial cells. This is a gland. The gland itself is separated from the rest of the prostate by this basal membrane. I'm hoping that when I'm moving the mouse over here, you can see the movement of the mouse because I'm trying to describe the different aspects of the prostate. Inside the gland itself, there's the lumen, and the lumen is important because it's really what carries the semen fluid, which is what these prostate glands are supposed to be carrying, right? So this is what they're trying to do. There's a number of cells that this model is not going to consider. The complexity of the cancer ecosystem in the prostate is well beyond what any of my models could tackle, but we're just trying to at least add some of the complexity which would allow us to sort of ask interesting questions that simpler models wouldn't be able to, right? So you have all these cells. These are normal epithelial cells. They're just doing their thing. They're surrounded the lumen. The lumen right now obviously is just an empty space, but is supposed to carry fluid. You have a basal membrane separated from the rest of the prostate. This basal membrane is very important because what happens inside the lumen is not impacting what happens in the stroma. The stroma itself, this environment, has basically two types of stroma, which is just like a very crude characterization of the real sort of cellular heterogeneity of stroma, which includes monocytes, myeloid cells, smooth muscle cells, fibroblasts, and so on, right? So you have static stroma, which is just a catch-all phrase for fibroblasts and other similar-looking cells. And then you have modal stroma, which is a catch-all phrase for monocytes and macrophages. Yes, thanks, Lorenzo. I'm glad that you can see the pointer. I think that given the audience, you all know what an agent-based model is, so I'm not going to spend any time describing them. Please do correct me if I'm making an assumption that is not entirely right, but I feel like it's going to be a lot easier if I start explaining how I implemented this ABM instead of describing what an ABM is. But one thing about ABMs compared to other modeling approaches is that they give us this small window with high resolution in terms of space and time, where we can actually track how the progression of disease is initiated and continuous, rather than something that allows us to track entire populations in a more clinical way. We have a very small window into the disease, but it's a window that has a lot of resolution, and it gives us a lot of detail that most other mathematical modeling approaches wouldn't be able to give us. We're using an agent-based model approach that is based off the HCA paradigm, and the HCA is basically a way to say hybrid discrete continuous automata model. So we have a continuum part, which we'll describe later, and we have a discrete part, which is what most people think about when they think about agent-based models. It's basically a cellular automaton where every single agent is a cell that follows a set of rules. And these flowcharts are just basically a very crude characterization of the rules that these cells follow over time. And as you can tell, we have tumor cells. What we have mostly is other cells. And we are very careful about modeling the normal behavior of these cells, because these normal cells is something that we can understand, we can model, we can parameterize, and we can validate without having to understand tumor cells, because every person who has a prostate will have normal cells doing the normal things, easy to measure, relatively consistent. And we can make a model that captures this normalcy in ways that are really rigorous. So then we can actually figure out what tumor cells would do to this ecosystem once we have parameterized the rest of it. Now, the discrete part was these agents, and these flowcharts describe the behavior of these agents. There's a continuous part, and this continuous part describes the tumor microenvironment in regards to things like signaling. Transformer growth factor beta, TA-beta, is a really important signal that is sort of secreted and listened to by the cells, normal cells, but also by tumor cells later on. And it's super important because it maintains homeostasis. It allows for the prostate to be under sort of control without proliferating much, without dying. So this TA-beta in normal cells, normal epithelial cells, is just something that allows them to survive. So it's a survival mechanism that's super important. Tumor cells would still be subjected to the whims of TA-beta, but they also limit proliferation, and tumor cells learn how to ignore that part. So in a normal situation, TA-beta means don't proliferate unless you really, really need to, but you're allowed to stay alive. Then you have these matrix-degrading enzymes, which are basically these enzymes secreted by tumor cells. And what they do is basically they degrade the basal membrane, they degrade the extracellular matrix in the tissue of the prostate, and allows for the proliferation of prostate cancer cells and for the invasion from within the gland to the outside. And then you have the basal membrane and the extracellular matrix themselves. Those are going to be impacted by the matrix-degrading enzymes. And it's important because they basically create this environment which maintains homeostasis. It's something that stromal cells will be maintaining in terms of long periods of time. So basically, they can respond to damaging and degradations of the extracellular matrix and the basal membrane. And in this particular model, we decide to model them as if they were the same thing, such that, as you can see in this screen, if you look at this window here, that's the basal membrane and the extracellular matrix. The concentration is given by colors. If you have black, that means absolute lack of it. If you have white, it means top concentration of something. And what this image shows here is that we have a normal small value of something for most of the domain of this cellular automaton. And then you have these three rings with high concentration. So high concentration of this signal means there's a basal membrane. Low concentration of the signal means there's an extracellular matrix which is there and is sort of slows down tumor proliferation and it slows down the secretion of molecules. Then you have these molecules. At the beginning, the matrix-degrading enzymes that you can see here is non-existent because only tumor cells produce them. And then you have the transforming growth factor beta over here, which, as I said before, is secreted by normal cells constantly all the time, is something that maintains homeostasis of the epithelial layers, because when epithelial cells, normal cells, non-transformed epithelial cells, so non-tumorous ones, look at this, they think if there's space for proliferation or proliferate, but most of the times this is going to inhibit my proliferation and also it's allowed me to keep alive. So if there's TGF-beta, epithelial cells are happy. If there's no TGF-beta, they'll die. And finally, this screen over here is, well, you can see the dynamics of the different cellular populations in action. So all these universes exist in parallel, and the cells over here can see the transformer growth factor beta field over here, the NDE field over here, and the basal membrane and extracellular matrix layer over here. So these three glands, you can see three of them. You can see two different sort of colors, blue and green. The blue line is the luminal epithelial cells, and the blue line is a bunch of cells that are the basal epithelial. It's just epithelial cells, that's what they do. And then separated by a basal membrane that you can only see in this screen, but separated by them, there's the rest of the stroma, and you can see two colors, there's brown and there's black. And one is the motile stroma, which is basically these monocytes and macrophages, which follow gradients of transforming growth factor beta and repair the basal membrane and the cellular matrix when needed to. And then you have the fibroblasts, or the static stroma, as we call it in this particular model, whose job is to basically listen to changes in the extracellular matrix, to listen to changes in terms of signaling. They receive TGF-beta and they amplify the signal, creating gradients of chemostatic gradients that other cells will follow. So let's just look at one of the simulations. You'll see how in each of these glands, at a particular time point, six tumor cells will be initiated. The middle gland gets 4 tumor cells, and each of one of the other glands gets a tumor cell. You see, because it's red and it starts growing immediately inside the lumen of the glands. So you can see that. You can see in this screen the concentration of these matrix-degrading enzymes, which are secreted by the tumor cells. And their job is basically to sort of start degrading the basal membrane over here and the extracellular matrix. So you see that this yellow field here in the middle of each one of those glands gets darker and darker as time goes by, because the matrix-degrading enzymes produced by tumor cells start degrading them. And as they start degrading them, and as they start degrading the basal membrane, separating the inside of the lumen to the outside of the gland, you can see how transforming growth factor beta over here begins to leak out. You see, there is a lot of stuff going on here, but we have these models parameterized using really well-established data, just because most of the cells in this model are really well-known, well-established. There's lots of literature, there's lots of experimental data we can use. And the only thing that we have to spend a lot of time thinking about carefully is about the tumor cells themselves. But the rest, it was easy to get numbers for. And you can see these cells over here moving a lot. And the reason they seem to be moving really, really fast is because these simulations simulate times that are in years and in decades. That's because prostate cancer is a really slow progressing cancer. Prostate cancer is the kind of cancer that a lot of people get. Is one of the biggest killers in terms of males with cancer. Is the second biggest one. But if you only have prostate cancer and there's no evidence of metastasis, 90% of the patients survive. And the reason why that is the case is because it takes so long. It takes so long for the cancer to progress that in many cases, the patient who is already likely to be in the 70s or even older is likely to die of something else of old age rather than the cancer itself. So basically, it's not necessarily a very serious disease unless there's evidence of metastasis. But that's not the situation we're here. We're in a situation where the tumors are still contained inside the gland. And the interesting thing is, if you look at this T-beta leaking out, because it's leaking out because the basal membrane over here is being degraded very slowly by these metis-degraded enzymes, that means that T-beta is being listened to by motile stroma and by static stroma. The static stroma takes this T-beta, amplifies the signal, creates chemotactic fields that are then followed. And you can see these chemotactic fields over here and over here and over here. They are followed by the motile stroma, which starts congregating around where the T-beta is leaking the most. And as you can see here, it's generating a cellular matrix. And this cellular matrix itself slows down the secretion of MDs and is slowing down the diffusion of T-beta. So there is this really dynamic, very interactive conversation going on between the tumor and the stroma, which is right now in such a place where stroma is trying to sort of contain the tumor to make sure that is still within the gland. Obviously, the gland itself has a basal membrane that is slowly losing its power to keep the tumor inside. And the tumor cells are doing the best they can in order to escape from the confines of the basal membrane, because when they do, they kind of start taking over the prostate. And I'm just going to accelerate the process a little bit because I don't think we had that much time. So I'm just going to move this a little bit, but you can see how the motile stroma is here. It's really hard at secreting extracellular matrix. You see these white colors over here. That means that they are really producing a lot of extracellular matrix. But you can see now how at some stage the tumors manage to escape and they start coming out inside what is the main area of the prostate. So of some of the glands, they're really invading. At this stage, this cancer is no longer a pink cancer, a prostatic intraepithelial neoplasm, which is like a non-very serious kind of prostate cancer, into something that is a lot more concerning. It's beginning to invade the tissue itself. And what we did is to ensure that we could explore a number of scenarios. Like you have a model like this, and then you can start playing this game, which is like, we know how the prostate looks like. In some cases, in younger patients, we have a lot of stroma. In older patients, that stroma is a lot less active. There's a lot less fewer fibroblasts. There's a lot less monocytes. So can we explore different scenarios where you can sort of, this here, 40-40 means that we have a lot of stroma that's static, a lot of stroma that is motile. This scenario here is when you have a lot of static stroma, but not a lot of motile stroma. This scenario here is when you have a lot of motile stroma, but not a lot of static stroma. And here, 10-10 is when you have very few motile cells and very few static stroma cells. Now, this number 10-10 is just basically the amount of the domain that is taken by a particular population. So 40-40 would imply that 80% of the domain at the beginning of every simulation is taken by stromal cells, and 10-10 would imply that only 20% of the entire domain in this simulation is taken by stromal cells. And we can play with the phenotypes of these tumor cells. So what happens if you have a rich stroma prostate and a poor stroma prostate? And what happens with these tumor cells, which we're going to characterize by two different things? Obviously, you look at the model, there's two different aspects that the prostate cancer cell can do. It can generate T-beta, which we know is very important because it maintains survival, and it can change the speed at which they generate matrix-degrading enzymes. And what we find is that this transformer growth factor beta, which is if you talk to any prostate cancer biologist, they'll tell you T-beta is super important. We know that tumor cells upregulate T-beta. We know that when they metastasize to other organs, they have this huge upregulation of T-beta. We know that increased survival. We know that it's going to help the tumor grow faster. Well, our simulations suggest that that's not necessarily the case, and that by changing T-beta in almost every situation, you don't see a particular change in the kind of output that comes from this. And the outputs are basically either green, which basically means the tumor dies, yellow, which basically means the tumor grows but remains within the confines of the gland, or red, which what happens when the tumor cell grows, takes over the prostate, right? So we can see is that we can assume that the seeding cells in each one of the simulations can have all kinds of phenotypes where they produce either a little bit of T beta and a little bit of MD, or they produce a lot of T beta and a lot of MD, and every single combination in between, in environments where there's a lot of stroma and environments where there's not much stroma. And it doesn't really matter what you do. In general, the rule is that producing more T beta doesn't seem to play much of a role. Whereas the speed by which they produce these metastatic enzymes goes a long way to explain what kind of progression the tumor is going to get. So for us, was very important. And I think, yeah, no, I don't have the validation here. And that's because I wanted to make sure that this presentation was going to be about the models and not the experimental data itself. But we actually did validate the results to the surprise of our experimental collaborators, Simon Hayward and company, who were back then in Vanderbilt University and now the University of Chicago. They found out that if we could actually engineer mouse models with prostate cancer cells that would overexpress T beta, that would not change the size of the tumors, that wouldn't change the ability of the tumors to actually grow or be extinct. It wouldn't change anything. Whereas it would actually change the ability of these tumors to sort of interact with the prostate, with the stroma in ways where our model captured that. So the amount of collagen generated in areas where there's extrapolation of T beta correspond to what we saw in simulations as well. So that was an interesting result by itself. But what we have here is just basically the early stages. Later on, we decided to say, okay, we now understand better what the dialogue is between prostate cancer cells and the stroma when the tumor is just initiating itself. But obviously, the situation gets a lot more serious when the tumor cells can escape and start invading. So at that stage, one of the things you might actually look at is how much reactive stroma a tissue will have. Reactive stroma is just basically this stroma component, these fibroblasts, these monocytes, these macrophages, these smooth muscle cells. And obviously of them, fibroblasts are probably the most important ones. They are the most abundant stromal cell that you can find in the prostate. They play a huge role. They're really involved in maintaining homeostasis. They are very involved in maintaining when there's any wound or any change to go and repair whatever damage has been done, right? So that's important. And one of the things what happens with age is that a lot of these fibroblasts become senescent, a lot of these fibroblasts become faulty, a lot of these fibroblasts become reactive, which basically means that they react to cancer cells in ways that might not hinder, as we've seen before, but potentially promote the growth of tumor cells. And because we are very interested in how this impacts cancer evolution, we decided to keep modeling this. Now, the model itself did not change a huge deal. What changed is the size of the domain, because we now wanted to explore dynamics that have an evolutionary turn to it. What we did is take some images from pathology, draw some glands over here that instead of being circles have morphologies that are slightly more realistic in terms of how they look like. Not that they change a huge deal, but from the point of view of presenting things that are more believable, when you talk to biologists and clinicians, just having shapes that look more realistic, it goes a long way in making them believe the model simulations. And this looks like a really complex prostate over here, but what we did is just create a much larger environment and just basically copy paste the same pattern in a number of places as to have all these glands. And what we do now is, instead of just letting the prostate in the gland and just wait, we just initiate it in ways where it's easier for the prostate to escape from the gland, and then explore what the dialogue between the tumor and the stroma is once the tumor is already growing inside the prostate itself and outside of the glands. And now we have this more complex model, because instead of having simulations where, then again, the model themselves is not different, but instead of having tumor cells that have a phenotype that is shared by every single tumor cell in a simulation, now in every simulation, when tumor cells divide, they might produce offspring that is different. So before, when we initialized a simulation, we decided, well, the simulation itself will have this phenotype in regards to production of T beta, and this phenotype in regards to production of these matrix-degrading enzymes. But now we're just going to let the environment select for the phenotypes that are better suited for this simulation. So you start the model with a particular phenotype, and every time a cell divides, it might change the amount of the NDs and the amount of T beta that this cell produces. And that obviously will have a lot of impact on the simulation themselves, because now different elements of the macroenvironment will select for different phenotypes. So now these simulations are very colorful and they're very pretty. And as you could see before, you can basically see in different screens, different aspects of the simulations. Now, this is an environment that is RSD1, which basically means the amount of reactive stroma, the amount of stroma that potentially can help you because it's senescent or not properly functioning, is very low. And you can see the tumor has grown to this size over here. Remember this because we'll see a simulation later on where the environment is more permissive, but this is not a permissive environment. So this is the size of the tumor. This is the amount of growth factors that the tumor can see and can take advantage of. These growth factors are secreted by the tumor itself, or they're secreted by the stroma. If the tumor lives in a senescent stroma facilitating environment, then fibroblasts themselves might secrete the factors the tumor needs. And you can see here the tumor phenotypes, where blue colors means the tumor is not very aggressive, it doesn't produce many growth factors, and paler colors mean that it's producing those factors, right? So what happens when the environment is more permissive? So you see the colors here are a lot redder and yellower, which basically means there's a lot more of them. But the colors here about the phenotypes of the cells themselves, how much they change over time, they are a lot bluer. So let me just go back because I think this is important. This tumor is much bigger than this tumor. So this tumor it doesn't grow very much. The environment is not helping. But this tumor now, where there's an environment that's allowing the tumor to grow because there's a lot of growth factors, it grows much bigger, but it doesn't need to change, right? So because the environment is very permissive, there's not a lot of strong selective pressure from Darwinian evolution to select for cell types that produce lots of growth factors. So paradoxically, and this is something that is a very important message to your clinician, is that you can actually have a tumor that grows very fast, that grows very aggressively, that's made of tumor cells that are not very aggressive because the environment is permissive enough that the tumor doesn't have to evolve a very aggressive phenotype in order to grow. Whereas you can have a much smaller tumor, as we saw before in RSG1, where the tumor doesn't seem to grow very fast. And the reason it's not growing very fast is because the environment is not permissive. And because it's not permissive, it needs to evolve. There's a lot of strong selection in the environment. For tumor cells, whenever they divide, there's a strong selection for the ones that actually produce growth factors and NDs that allows them to sort of grow faster and more aggressively in this environment. So the tumor cells themselves, you could take them away and look at them under the microscope. They look very aggressive. Yet overall, collectively, and this in turn something I think this audience will understand easier and more intuitively than others, collectively, the tumor doesn't look very aggressive. Individually, though, it's a very aggressive tumor. So which one do you have to worry about? So I don't know how many of you work with clinicians, but you work with clinicians, one of the standard ways to measure a patient's sort of progression and perspective in terms of the diagnosis is the Kaplan-Meier lines, which basically is starting from 1, which basically means at the beginning patients just survive for a period of time, but as time goes by, every drop means one patient of the cohort dies, right? So over time, the more aggressive a cancer is, the more that you can see these drops being like earlier in time and steeper as well, right? So you can see the blue line here dies for tumors where basically the patient is by clinician determined to be very unaggressive, very passive. And as you can see here, these patients actually sort of drop faster. Well, you know, if you're a prostate cancer doctor and you're a pathologist, what you look at is the shape of the cells. You don't look at the tumor in overall. You just look at a particular tumor cells. You look at the shape. You look at how they actually look on the microscope, and you make your decision based on that. They use something called the Gleason score, which is that when you're a prostate cancer patient, they take a biopsy, they look at a bunch of images, they take 2 that are representative, they score those 2 images between 1 and 5, 1 being like really not aggressive, to 5 being incredibly aggressive. And then they mix these 2 numbers, right? So you have a 2, that basically means you have no cancer, and you have a 10 or a 9, that means that you have a really aggressive cancer and your prognosis is very poor. Unfortunately, or actually, you know, to be expected, most patients that have cancers that to be worried about don't have 9s or 10s or 5s or 6s, they have 7s. And those 7s are really hard to know what to do, because sometimes there's no better treatment than to just wait and see what happens with the cancer. So basically using reactive stroma to further determine what kind of patients we're talking about, we can further determine and classify these patients. And we found that our classifying them this way by actually using a combination of the Gleason score, which is what prostate cancer doctors traditionally and conventionally use, is the gold standard, together with metrics of reactive stroma, which are also easy to get, easy to apply to the same samples they already get. It's much easier to further stratify patients that need further attention when they have like this harder to determine Gleason scores like 7s. So obviously, when it comes to prostate cancer, as I said before, like most of the cancers, you can be concerned well earlier as soon as they are detected. But when it comes to prostate cancer, because of the kind of patient that gets prostate cancer, more often than not, not always, but more often than not, we're talking about individuals who are in the mid-70s and further. And more often than not, it's a cancer that takes a long time to grow. So often you don't need to worry too much about it. And actually, one of the preoccupations that doctors have when it comes to prostate cancer is not how we treat it, but more importantly, should we treat it? And determining which one to treat is hard. Now, the moment it goes from a cancer that is just inside the prostate to one that's metastatic, the situation goes from should we treat it to for sure we have to treat it, but the prognosis is really bad. So trying to understand metastasis is really complicated, but it's also crucially important, because if you have prostate cancer that has no metastasis, your chances of living with it and dying of something else are 90%. And if you have metastasis, those odds are reversed. And then you become a kind of patient where 90% of the chances are that you'll die because of the disease. So that allows us to basically think of this as tumor cells that come from the prostate and arrive to a completely different environment. Now, this is a completely different model in terms of the fact that we actually build it from scratch. The other two models are basically iterations of the same model. This one is new, but it's also based off the same HCA paradigm, which basically means still a cellular model where every single cell behavior is given by a flowchart and encoded as a set of rules, and where there's a number of environmental variables and macroenvironmental features that are encoded in terms of partial differential equations that are discretized and then applied to different grids that are combined together to create this combined agent-based model. Right. So before in the prostate, we have these glands and we have these stall cells that fibroblast or monocytes. Now we're actually going to look at different populations. We have osteoclasts, mesenchymal stem cells. There's a complete different ecology. And what would determine if prostate cancer cells will grow in the bone is whether these prostate cancer cells that have basically intravased into the blood circulation, circulated in the blood for a while, survive the immune system there, managed to find a place to actually, like in high pressure, get outside and then grow somewhere else, managed to actually have the right phenotype to interact with those other cells over here. This is very important. It's also really hard. Most prostate cancer cells will die before they actually manage to get into the bone. And the few that manage to get into the bone are likely not to be able to do very much because they arrive to a very different environment than the one they come from. So yeah, this is basically just reiterating. Arturo is the postdoc in my group who did this work. Now he's a faculty member at the University of Roehampton in the UK. And Leo Cook is an experimental modeler who worked with Connor Lynch, who is now an assistant professor at the University of Nebraska-Lincoln, or Omaha, actually, Omaha. And we came together to create with this model. So when it comes to prostate cancer, almost any kind of cancer in the bone. And bone is one of the favorite organs for mold metastasis to occur, just because the organ is such a large organ, and the marrow is such a good place for tumor cells to grow. There's space, there's nutrients, and also there's all these cells who are doing things like bone remodeling and bone homeostasis, secreting factors that often can help tumor cells grow and establish themselves, where in other organs it will be a lot harder. So the vicious cycle is just basically tumor cells coming in and taking advantage of all this dynamic organ, which is the bone. And now I didn't know how dynamic the bone was until a few years ago. But now I realize that it's an organ that is remodeled every few years. Every part of your bone is different. And the reason is because every time you go for a walk, for instance, there's going to be microfractures and things that need to be repaired. Every time your body needs calcium, the bone is the place where there's like a bank of calcium that the body can take care of. And these bone cells, like osteoclasts and osteoblasts, are the ones that make this possible, repairing the bone and absorbing and depositing calcium when the body needs that. As I said before, and we actually published this research in a number of places. You can go and take a look. Tumor cells, as well as stromal cells, are sort of governed by this equation, by these flowcharts. And this is a simulation where there's no cancer. So just don't look for cancer in here. This is the white space, the bone marrow. This pink space is the bone. This cell here is a MSC, which is a stem cell that is there maintaining bone homeostasis. And then you see very quickly loss of blue cells and red cells and other cells. And these are the osteoclasts and osteoblasts, which basically come in and destroy bone and produce bone, which is what you have to do whenever you want to regenerate bone that needs to be repaired for whatever reason that might be. And the way they orchestrate the behavior, the way that bone actually gets remodeled, the way the bone actually sort of gets homeostatic is through interaction within these cells through signals like TA-beta, which we talked about before, and which we now know that prostate cancer cells generate themselves. Rank ligand, which is another sort of important signal, which basically recruits these precursor osteoclasts that will come on and to a bone, and other bone remodeling factors, which we just cluster together. So you can see the concentration here. You can see this recruitment of osteoclasts through rank ligand, which means that these osteoclasts come in and fuse together, creating this red line, which is like a fused osteoclast, which goes and resorbs bone, destroys the bone, and the osteoblasts, which come behind it, differentiate when the osteoclast dies and start secreting bone. Now, this process has not been hard-coded. We basically create an ABM-based model where we allow for the cells to interact with each other in ways that are realistic from the point of view of the time scales, from the point of view of the signals that are exchanged for the orchestrating of the process, from the point of how far the signals diffuse, how fast the signals diffuse. If you put all the things that we know very well, because the bone is a normal organ that is well characterized, if you take the parameters that we already know very well, and you put them together into this model, it doesn't take a lot of effort to get something where all these cells automatically by themselves, sort of emergently, create this process of bone remodeling, by which over long periods of time, you can see how the bone goes back to a degree of homeostasis. And having a good understanding of his homeostasis is important, because then you can put a tumor cell, which you can see here in the middle is this cell over here, and see what happens, because this tumor cell is generating transformative growth factor beta, which is usually used to orchestrate bone remodeling, which basically means that T-beta is expressed in places where these normal cells, these osteoclasts and osteoblasts, are not expecting them, which basically means that this bone remodeling process just goes haywire. You have all these remodeling sites going on at the same time. And every time there's remodeling, there's these factors like rank ligand and these BDFs who are generating lots of growth factors for the tumor cells. So tumor cells grow, which means they overexpress more T-beta, which basically means that the bone remodeling process gets even more crazy, which basically means there's more bone remodeling, which basically means tumor galore. So that's the situation where we're in right now. And because we have this model, then we can begin to explore what is the role of different treatments in the clinic and what they can do to it. I'm not going to spend much time discussing about this because, as I said before, I don't think the goal for me in this presentation today is to sort of give you a clinical understanding of what this model tells us. It's more the fact that having models as simple as this, relatively simple, I'll say, allow us to have this very close, very high resolution view of a very small segment of the cancer. Obviously, the cancer will be billions and billions of cells, whereas here we're actually only modeling tens of thousands of them. Obviously, if you know the work of James Glazier, you know the work of Paul Macklin, especially with Paul's physical, you can sometimes generate these really complex simulations with really high efficiency of huge amounts of cells. And this is a much more modest model when it comes to how many cells can we simulate. But it doesn't matter how good your agent-based model is, it's going to be really hard to model something that is the entire burden of a tumor in the entire sort of length of a bone. Bones are huge organs, and it wouldn't make sense for us to try to understand with an agent-based model in 3D the entire sort of length of the bone, right? But there's a lot of insights, there's a lot of understanding that comes from having this very small window with lots of resolution in terms of time and space, what happens with the bone and what happens with the prostate and how the tumor cells, and we can explore all kinds of phenotypes due to the bone, how they take advantage of this bone environment, how they take advantage of this dynamic process, which is called bone remodeling, which is ongoing. And you can assume there's at any point in time, there's bits of your bone that are being remodeled. And also, you know, by understanding not only the homeostasis of the bone and what makes the bone and the process that determine bone health and how tumor cells can actually take advantage of this, then we can see the impact of treatments not only on the tumor cells themselves, but in the bone microenvironment as well, and the indirect impact of many of these treatments in the efficacy on the cancer itself. Let me see, I have a few. Oh yeah, okay. Thanks, Jane, for sharing all these papers. Now, very briefly, now I'm going to move on to multiple myeloma because that was the next project we decided to work on. This is another group of postdocs in our groups. In the case of Connor Lynch, the bone biologist I told you about before, Brian Bishop, which you can see in this picture towards the end, is the experimentalist who has helped, and Anna Miller, who is now a senior scientist at Certara, which is a PKPD company doing great modeling in North Carolina. We decided to understand not only what happens with prostate cancer, with multiple myeloma as well, which is as opposed to prostate cancer, not something that initiates in a different organ and comes to the bone, but it's a kind of cancer that grows in the bone, for which we have treatments that do impact the cancer, but not in ways where actually we cure the cancer. So basically, we have things we can throw at patients who have multiple myeloma. Those treatments have an impact for a period of time, but by and large, these treatments don't cure patients from the cancer. So it's definitely something for which we need to come with better ways to understand what's going on and how we treat them. And this is slightly, we created the model from scratch this time using a platform called HAL, which is a bit like PhysiCell and CompuCell, is a way to slightly more easily create agent-based models. Just as our colleagues in Indiana, we found out that creating these tools is very important because instead of having to start these agent-based models from the scratch, every single time you have a new group of postdocs trying to come with a new project, having these libraries not only accelerates the creation of these agent-based models, but also because there's a community of people working together in these tools. It's much easier to learn from other projects, incorporate ideas from other projects, and potentially even do modular models where different sort of parts of different projects can actually jive together. So this is a different model, also from the scratch, using HAL. You can see the trabecular bone here. This is still the bone marrow, same colors. We created a bone that looks slightly more jagged because this is how trabecular bone really looks inside the bone marrow. It's not like a nice, smooth surface. It's a lot harder than that. And we made sure that this time bone remodeling would be able to go on for long periods of time and still capture this health of the bone. Because as I said before, it's very important for us to be able to capture homeostasis. Every single model I've shown today can capture for long periods of time the dynamics of normal healthy cells, epithelial and stromal cells interacting with each other to maintain organ homeostasis. Because once you understand this, you have created a benchmark and a foundation over which you can begin to understand what tumor cells are going to be doing to tissue. If you don't have that, if you start having tumor cells growing in a vacuum, that's great for certain questions. But for us, the more interesting questions, which are the ones that involve stroma, you need to be able to understand whether your normal cells are doing what they should be doing. And as you can see here, we can have simultaneous elements of bone remodeling going at the same time in this piece of bone. Now, usually you wouldn't have that many of them going at the same time, but you can see how all these things can go at the same time without impacting each other. So basically, we can have like a lot of stuff going on. We can really sort of throw a lot of things to this bone, and this bone will be able to recapitulate homeostasis in biologically realistic ways. There's a lot of biology that I'm not going to tell you about how we actually drove this model, because there is a lot of assumptions these agent-based models need to make. Obviously, we were more than lucky that Ryan actually had this beautiful energy mice immunofluorescent mouse model, and this cell line model, which we could inject and see the impact of these cell lines in the bone. So if there's no tumor, then obviously the bone is normal. And if you put tumor, you can see how it destroys the bone. We can talk all kinds of cells, tumor cells and non-tumor cells. We can try to see every single treatment as well. So we can see how different tumor cells survive near the bone, how tumor cells survive when they're not near the bone, and we can then begin to study heterogeneity. So here is bone homeostasis. Here we have tumor myeloma cells. These myeloma cells in this particular simulation, they just are the same. So every single tumor cell resembles the other. They have the same rates of growth. They have the same rates of death. They have the same sensitivity to treatments that we'll see later. Right, so that's not important right now, but basically you can see that obviously when there is no tumor growth going on, the bone is just homeostatic. When there's tumors, these tumors interact and take advantage of bone remodeling like they did in prostate cancer, but this time, as opposed to being osteolytic, they're osteoblastic. They actually lead to, in being osteoblastic, which basically means more bone is created, they're osteolytic, which basically means bone is destroyed over long periods of time. This is what we expect for multiple myelomas. So the fact that this emerged naturally without us having to change the model substantially was quite reassuring. Now, when it comes to parameterization, because we have access to a lot of mouse model data, experiments that were done exclusively to sort of drive the parameterization in this model, we could have comparisons between the outputs of the model, not only in terms of the amount of volume of the bone, in terms of the tumor cells themselves, but also every single other population, like the stem cells, the precursor osteoblasts, the other osteoblasts, and so on. We can compare qualitatively every one of the populations, and because this is a couple model where every population impacts each other, we have a degree of assurance that this model is behaving in a biologically realistic manner, because it's very hard to get all these dynamics going on at the same time. And for each one of these populations and sort of have a comparable comparison with the in vivo situation without at least leading us to believe that the model is reliable. I don't know if I have a lot of time. I suspect that this meeting needs to be done by around 4 Eastern. So I'm just going to wrap up nicely here, I suppose. But we have treatments, and we now have ways to parameterize these treatments. We wanted to explore the role of external protection, and we found out the environmental mediated drug resistance. So when the environment itself, the tumor cells can derive protection from the growth factors generated through bone remodeling, when they can get protection from stromal cells directly by physical contact. This EMDR leads to patterns of resistance from conventional treatments that are very different and that lead to more intratumor heterogeneity. Once again, the goal of this presentation is not to really show you clinical results, is to show this progression where we start from early stages of growth in the prostate, growth inside from the prostate glands into the prostate itself, metastasis to the bone, to show you how all these agent-based models with the right parameterization and the right sort of calibration through homeostasis and through sort of experimental data can allow us to gain new insights in how the disease progresses, changes, grows, metastasizes, and responds to treatment. And with further ado, I'll just obviously thank again my group for all the work they've done. I want to reiterate that we have a special issue on cancer, collagen evolution. Obviously, as an editor, I'm more than keen to see anyone in this particular audience who might be having anything that might be relevant. I know that not every one of you is working on cancer, so it might not be necessarily something for you, but if you know anyone who might be interested, please let me know. And if you have PhD students who are interested in mathematical oncology, we do have a program here at Moffit. It's one of the few places where you can actually work with clinicians, experimentalists, and math modelers and get a PhD while you do that. Thank you very much.
That was great. Thank you very much, David.
Yeah, I'm sorry I had to rush things a little bit, but yeah,
that's okay.
I have a lot to present, and
that's okay. That's okay. Are there any questions?
Yeah, maybe. Can you hear me? Yeah.
Yes,
yep.
yes.
Lucas, go ahead, Lucas.
Thanks. Yeah, I'm not an expert on cancer research, but I found some of these simulations you showed very interesting. In particular, the one I think it was early in the presentation where you showed that you took some tissue data, I think, and then transferred it to your to your ABM. And is this correct? Right? Like I think you took some.
Is it over here?
Yeah, right. And this one. So I was wondering, that's based on real data, right? And then you kind of used it in your ABM model. So I was wondering, is it possible to model something within your current framework that is resembling, let's say, a real like tissue model that is, kind of reminiscent of something that you would observe in an actual patient? And is it possible to run these simulations on like normal computing equipment, or how do you simulate these kinds of ABM implementations?
These are great questions, and I'm happy that you're asking them. Now, when we started this project, and obviously you see the date of the application is 2020, but we started well before that. We went from simulations that look like this to simulations that look like this, and you can see that the realism, the shapes of the glands, all this has changed substantially. When I started this transition for the project, I took pathology images from the pathologist who is one of the co-authors of this paper, Gustavo Ayala, at the Baylor College of Medicine, and I did something that is time-consuming, but it's very likely. So you basically take an image from pathology, you sort of black and white it, you threshold it, and then you use that to generate like, okay, so every one of these black dots is basically a gland. So I'm going to sort of assume that this is where the gland starts. There's going to be epithelial cells all surrounding these things. And you can use that potentially. You have enough time to run a simulation. We have a cluster here at Moffit. I don't know how common it is for hospitals to have a computing cluster, but we have a cluster of modus means here at Moffit. And you can run these things in a realistic amount of time. But the reason I have this sort of big thing here is I don't want you to think that these models are necessarily the kind of model that you want to use in order to have to generate clinically interesting predictions. For me, the reason I created this model is not to go on and tell the doctor, give me a patient, give me some profile of the tumor cells, and I'll be able to generate how exactly the tumor is going to grow in the patient, because I feel like this will require a lot more work. You need to be a lot more careful. I just want to understand the biology, because we feel that there's a lot of biology that's not really known about these early stages of cancer. And there's a lot about the dialogue between the tumor and the stroma that has not been used therapeutically. That's the only concern. And going from this really artificial to slightly more realistic image, initial configurations like this allows me to convince doctors, allows me to create use more realistic shapes, so the model outputs are not an artifact of the particular shapes I have used. But even this much larger simulation is still a tiny fraction, and it's a 2D simulation as well. Would it be able to allow me to, in confidence, to go to a doctor and say, given this treatment, given this patient, given this tumor, this is how big it's going to grow, and this is how much it's going to change? I feel like that will require a lot more work. So my idea is, when I started, was exactly what you just said, but I think that my goals became more modest the more I understood the limitations of the approach.
Okay, yeah, thanks. That's very helpful. Also, the comment on that it's useful maybe to use these kinds of simulations to convince doctors that it's not just some kind of sensitivity analysis on these simulations.
Yes, that's right. Yes.
So that's very helpful. Okay, thanks.
Thanks for your question.
Lorenzo?
Well, thank you for the presentation. It was very nice. I really like all the project you've shown. Well, I have one that comes from my background, and which is if you have considered the vascular compartment in some of your simulation, or if you have given some thoughts of that, if is useful or worthy this to implement that, or if you already done it. But that's one.
Yes. So that's an excellent question as well. I mean, if you it depends on who you're presenting these models to. Some people might think this model is too simple, and some people might think this model is too complicated. And I feel like the audience here, because you guys are ABM-based modelers and multi-scale modelers, you understand that there is a huge and fine balance in terms of how much complexity you want to put into these models. It's very important to get it right. And we all strive to make sure that we put the sort of things that are important to our question, but no more detail that would actually obscure what comes out of the simulations, but also not to make them too simple just because it's more mathematically tractable. You want it to be sort of complex enough, right? And angiogenesis is such a huge, important aspect that I think is a reasonable question to ask. Why haven't we not asked in this model the role of vascularization? It's important in this model, and it's even more important, I believe, in this model, and very important in this model. This is important because, you know, a lot of the cells that are recruited to the process of bone remodeling are recruited through vasculature. So where vasculature is important. How the tumor is going to grow depends on resources. Vasculature is important. Now, if you want to do justice to the way the vasculature is in a model like this, you can go and say it's going to be too complicated. So I just assume that there is oxygen plenty and glucose plenty, which is what I did here. It's a big assumption and it's not true, but I'm just hoping that it doesn't hurt what comes of the model very much. Or you can actually sort of model angiogenesis properly, which I have friends and colleagues here at Moffit who have done it. And it's a really complicated process that involves the communications through BEF and HIF1 alpha. And there is also the role of tumor heterogeneity, because some cells might produce more HIF1 alpha and others will produce fewer. And the endothelial cells that guide the creation of vasculature and how this vasculature gets sort of physically constrained when the tumor grows around it, how much leakage there is. So you want to do it really properly. It's a lot of work, and I didn't think that we would have the time for it. If you want to do it very poorly, which is what we did here, you do what we did. In other simulations, what I did is the following, which is like I dotted the domain of the simulation with dots that are just representations of the vasculature going towards you. Basically, if this is a 2D simulation, you can imagine the vasculature going in this z-axis, and that nutrients, oxygen, and glucose diffuse in certain way coming from these dots. And then you can make a simple assumption saying, like, you know, we can easily capture that. We can easily make assumptions of how much oxygen and glucose from these things, how much that is going to impact tumor proliferation, the recruitment of these other cells like osteoclasts and osteoblasts, and potentially, you know, if a tumor cell grows around a vasculature, potentially say like this vasculature is not working anymore, and a tumor that is very successful in a particular area might kill the infrastructure is there providing goods. So leading to even more dynamism that I think will be very interesting. I think that might be one easier way to include vasculature without having to make the model even more complex than it is already.
Thank you
Obviously,
for that.
you have suggestions. I'm more than happy to hear them.
I have another one on another side, which is, I guess, this slide approach. The idea is, have you tried to put together your models? So, for example, you have bone in both of your models. Have you tried to put everything together to say, okay, we have standard ways of treating bone, for example, that we use a couple of times, and then we will treat prostate for another kind of disease, we might use again our model. So there is some reusability, or you are explicitly trying to kind of make it that way.
I've been thinking about that for the last year, so I haven't started yet, but those thoughts have really come to me because you could use one of the models to determine which kind of tumor cells succeed in the prostate. Those are the cells that are likely to metastasize. So you could extract those phenotypes from the prostate cancer model and put it into the bone cancer model. Then you can sort of narrow down the kind of cells that you should expect in the bone and see which ones grow there, and allow us to better understand. Because for certain cancers, we know that metastasis is something that occurs relatively early on. So a lot of people think that metastasis happens later, like you have a cancer and it grows for many years and then metastasis happens. Chances are that cells are seeded into metastasis from the very beginning, but most of these cells are not ready to metastasize successfully. They are wasted. So what transition does it happen? What does it take for these early prostate cancer cells to have whatever aggressiveness to evolve to the point where they can actually successfully establish themselves in the bone. And I feel like by actually having two models, one the prostate and one the bone, would actually answer the question very nicely. So your idea is great. And yes, I've been thinking about this, but you've been thinking ahead of me in the way that, yes, I have thought about this, but I haven't had time to actually sort of do something about it.
David, I have a question
Thank you very
about,
much. Thank you.
about
Okay.
five
Thank you.
slides. About 5 slides earlier, where you had the matrix degrading enzyme and TGF beta scenarios.
Here,
So
sorry,
a
here.
little bit farther back than that, because your summary slide. Keep going. That one dead center 14.
Yes.
So
So-
if I'm reading that correctly, you're saying that there is an intermediate level of matrix degrading enzyme, which is the bad scenario.
Yes, so when we wrote this paper, actually, this was one of the things that we spent some time discussing, because clearly that will catch in the eye of a lot of people, right? I did not do anything other than gloss over this time, because as I said before, I was just trying to put together models that usually I never presented together before, because I thought there was some value into that. But with you identify one thing I really gloss over very, very quickly. And the reason is because trying to explain this in a short period of time, I've been challenging. But you're completely right. T-beta, as you can see here, as you move from top to bottom, there's no huge difference from one scenario to the next in terms of what the impact is. It tends to not have a huge impact. The NDs, the matrix degrading enzymes that allow for the tumor cells to escape the glands to sort of invade the prostate cancer tissue, there's a Goldilocks value. You produce too much, that's bad for the tumor. You produce too little, you're confined inside the gland forever. So what's going on? We have simulations, and I just didn't put anyone here, just as I said before, because I didn't expect to be needing to present that, just because I didn't want to spend a lot of time discussing this particular result. But you're completely right. If a tumor, if you see any of these glands, let me just go back to the schematic here. So you have these glands, and I initialize each one of them with cancer cells, and they just grow. And each simulation, every cancer cell is the same, right? I change from simulation to simulation what the phenotype of the cancer cell is going to be, but every simulation has the same cancer cell. So if I simulate these cancer cells so that they produce whatever T-beta and whatever ND is, in most scenarios, what happens is the tumor starts growing inside the gland, start producing NDs. The NDs very, very slowly degrade the basal membrane, and the stromal cells come in, they try to plug the holes. Eventually, you wait long enough, potentially very, very long, more than realistically a patient would live for any reason, the cancer will escape. But sometimes a tumor might be very, very aggressive from one aspect and not aggressive enough in the other. That is, say that they produce not a huge amount of TGF-beta, which is something they need for their survival, and they produce lots of NDs. They could potentially produce so much ND that they degrade the extracellular matrix and they degrade the basal membrane so quickly that they might not have enough group effect. So as the tumor cells grow inside the glands, there's a lot of tumor cells, they all produce T-beta, there's a lot of T-beta, they're happy, they survive, they slowly degrade the basal membrane is degraded, they escape out. But if you produce with just a bunch of a small number of tumor cells enough MDs, you degrade the basal membrane, the TB you have is leaked out because the MD is being degraded not only the basal membrane but the extracellular matrix. The T-beta is a molecule that diffuses depending on how much ND and how much ECM is there. And it might actually go away before the tumor cells can actually go and take advantage of the space they created. So this is something that we have some weak validation for. So what we told that to experimentalists, they actually engineered that, and we don't have a way to mechanistically explain this, but we know that you can engineer tumor cells that produce MMPs, which is one of the proxies for MDS. MMPs is these metalloproteinases that sort of take away some of the ECM and realign the ECM. These tumors are less likely to succeed. And our explanation, given the simulation results, is that they are too good for their own good. They're just too efficient at degrading the extracellular matrix and the basal membrane for their own good.
Okay, and I also found it interesting that So in your bone metastasis model, bone remodeling stimulating tumor growth.
This one or the other one? Because I have two. One for prostate cancer and one for myeloma.
But if bone remodeling reduces growth factors which stimulate any tumor cells that have translocated to that site, so that would almost suggest that exercising is bad for a person with the bone metastasis.
Well, it's a lot more complicated than that, I'll say. But I mean, you're right in the sense that in many cases, tumor cells in the bone, metastatic ones, so not multiple myeloma, but prostate cancer, the bone metastasis. If you have tumor cells, many of them do really badly when they come to the bone. They don't know how to grow. They become dormant for long periods of time. And it's bone remodeling that can awake them and sort of make them active, when they wouldn't be active, maybe for even longer periods of time. Obviously, that's not going to change the fact that these cells were there and they were just waiting for an event to happen. You accelerated happening, but what's going to happen because your bone was going to be remodeled sooner or later. You just accelerate this. Now, we know that there's a lot more impact of exercising than just this. We know that it's likely to change your environment, like your stroma, how reactive it is, how conducive it is to help the tumor or not. As we see in these other simulations, let me go back to them. But you see, whether you have reactive stroma or non-reactive stroma, in part depends on your health. So you exercise, you might have an environment that is more conducive to cancer, and you don't exercise, you might have an environment that is more facilitating of the cancer, right? So you want to be healthy because your stroma, your ability of stroma to cope with challenges and to recover from insults and things that destroy homeostasis is going to be very different if you're a healthy person and a young person and potentially a person that exercises. So I'm hoping that you don't take this message not to exercise.
Yeah. Well, but usually, I mean, a prostate cancer metastasis is likely to be in somebody that's already in their 60s or 70s.
That's right.
It would be interesting to look and see if there's any, if for these bone metastases, if there's any localization around old fracture sites. You know, did the person have an old arm break or something? And because that changes both the trabecular and the cortical bone long term. I'm just wondering if there's anybody's noticed any, increased metastasis isolation there or less.
I think I did talk to Connor Lynch about this a while ago, and obviously I don't have any data to show you, but he was convinced that was actually the case, and in his mind he had read papers where, so take this with a huge grain of salt, but I feel like there's literature out there supporting that.